All right, before we finish up this chapter, let's do some review here. Which of the following is a property of a bacterial exotoxin? Is it A, a type of lipid associated with the cell membrane, B, released only when bacterial cells die, C, made of protein, or D, found only in gram-negative bacteria? Go ahead and pause the video. All right, the answer would be C. It is made of protein. All of these other ones are descriptors of endotoxins, which are found only in gram-negative bacteria. Okay, here's another one. Gram-negative bacteria can produce A, endotoxins only, B, exotoxins only, C, endotoxins and exotoxins, or D, neither endotoxins nor exotoxins. Go ahead and think about these. Pause the video. Okay, the answer would be C, endotoxins and exotoxins. So gram-negatives have endotoxins, right? The LPS in the cell wall, but they also have exotoxins that they can make, those proteins that they can secrete. Okay, in chapter 18.5, we're going to finish up by talking about viral pathogenesis. We're going to talk first comparing rhinovirus and influenza virus, talk a little bit about why one can kill you and why one won't kill you. We're going to talk about HIV and AIDS. We've talked about this before, but I want to tie it all back together. We will talk a little bit about human papillomaviruses and how they go about pathogenesis. And then we will talk a little bit about herpes viruses and latency, which is a fascinating process. So viruses, right, are different than bacteria. They are going to go about causing disease and causing damage in a different way. Viruses do have some similarities, right? They have to reproduce and things like that. But the way they go about it is very different from bacteria. For one thing, they're inert outside of the cell, right? Viral particles don't do any metabolism or things like that. Because of that, viruses don't really need nutrients or energy or do any sort of waste excretion. They just hijack the cell. So in a lot of cases, viruses actually want to keep our cells alive, right? There is the HIV/AIDS case where it is destroying the T cells, but that's a fine balancing act, right? If viruses destroy too many cells and kill the host, they have no way to reproduce and replicate. A lot of their tricks are going to be involved in prolonging cell growth and hiding from the immune system. Many viruses are very specialized to how they attach and trick their way into the cell and trick the cell into making more copies of them. In a lot of cases, we're going to talk about how they get in, what they do inside of the cell, and in some cases how they interact with the immune system, because of course they have to avoid it. So here's kind of like several different strategies they can use. Again, I don't need you to memorize this or anything, but I kind of want to lay it out. Some, like HIV, can infect immune cells. This can cripple the immune system, right? That keeps them around for longer. Others, like herpes viruses, have a latency phase where they hide from the immune system. As we've talked about, many viruses prefer specific tissues because they have the right things for tricking receptors or good growth temperature. So this tissue tropism is important. We talked about like hepatitis, only affects the liver cells, things like that. We will talk about altering viral antigens through evolution. Error-prone RNA polymerases, right? RNA polymerase makes a lot of mistakes, so we can have antigenic drift occurring. Segmented RNA genomes, like in flu, this can lead to antigenic shift. Cytokine production, they can actually increase or decrease inflammation depending on what they're trying to do with the cell. One very clever one is altering MHC I, which, remember, is the signal that a cell has an intracellular infection. And by definition, viruses are all intracellular, so they can mess with MHC I, as we see. Inhibiting apoptosis, so preventing infected cells from going through cell destruction. That leads to more viral particles being made, and sometimes even increasing host cell division, as we'll see with the papillomaviruses. Okay, so let's start by talking about two very common viruses that we've probably all encountered. We have rhinoviruses. These cause the common cold. We actually know of over 100 different what we call serotypes, which have unique cell surface antigens. So there are many, many strains. There's at least 100 of these different serotypes. That's really important, and we'll see why we can't create a universal vaccine against the common cold. They're not enveloped, they're single-stranded, positive sense RNA viruses. They make a protein on their capsid called VP1 that binds to the receptor ICAM1, which you may remember was used in extravasation in the inflammation response. That's just a side note. Rhinoviruses like cooler areas of the respiratory tract, so generally the nasal passage. This leads to generally non-life-threatening symptoms. We have runny nose and things like that caused by a release of bradykinin and histamine in that region. Your body, once a rhinovirus gets in, will make antibodies that will neutralize it, they'll bind to it and prevent it from getting into other cells. Those antibodies, though, will not recognize other strains of rhinovirus. So unless you get all 100 different serotypes, you're probably not immune ever, right? And that's very unlikely to happen. Okay, so this tissue tropism is one of the reasons why you don't die from the common cold. Influenza, on the other hand, has different infection strategy. So it's single-stranded, negative sense, segmented RNA genome. Remember, it's got those little pieces of its genome. So when it gets into a cell, and when two influenza viruses get into a cell, they can swap genomic bits around. That happens when, like, we have an avian flu and a human flu that go into a pig and swine flu or whatever, and they recombine with that antigenic shift. The influenza virus tends to infect the upper respiratory tract. In there, it actually kills mucus-secreting ciliated epithelial cells. So basically destroys your body's defense against it. Remember, mucus can catch viral and bacterial particles. You kill the cells that secrete mucus, they can now get into the cells below. So that infection can spread from the upper tract down into the lower tract and actually start destroying bronchial and alveolar epithelial cells. This is going to reduce the ability of the infected individual to exchange oxygen and CO2. So highly virulent strains can do this enough that it can actually kill someone through this process. So this is basically reducing your ability to breathe. And as we know, we need oxygen for that terminal electron acceptor, right? So we've got to have that. The symptoms that we experience, that lung stuff, is through tissue damage from the virus itself. We also get general things like headache, muscle ache, things like that. Those are all triggered from the microbial associated molecular patterns that the toll-like receptors detect. When it does this, when it's killing those cells, you're actually at greater risk of a secondary infection. So some other type of things like bacterial pneumonia or something like that could come into those weakened cells and kill you that way. So we have about 36,000 deaths from influenza in the United States every year, whereas from rhinovirus, I'm going to go out and say there are very few, if any, that occur. So why are these different? Well, in the rhinovirus, we have that antigenic variation, 100 different types, but that's a result of what we call antigenic drift, where slow errors in RNA polymerase lead to different ones. The different tissue tropism in rhinovirus leads to less damage that can kill us. The antigenic shift, that large-scale recombining of influenza viruses, can lead to new strains that are dramatically different. So they avoid the immune system that way. And historically, this leads to a flu pandemic about every 30 years. So the flu, right, very important that we stop its spread, because pandemics can be very devastating, particularly when we have large-scale recombination there. So that's why when we look for influenza vaccines, we actually look in the southern hemisphere because their flu season is offset. And so we look there and see what's prevalent, and we assume that that might make its way to the northern hemisphere. And so we make our vaccines against that. Although it doesn't always work perfectly, it's an educated guess at that point. So that's why we have yearly flu shots that we can use to provide immunity to the new strains. Whereas the rhinovirus, they're just out there and around. It would be very difficult and very costly to create vaccines against something that is at most generally a mild annoyance, right? So very different viral diseases caused by very different activities that are occurring. Okay, let's talk about HIV/AIDS. So HIV, human immunodeficiency virus, we have about 30 to 40 million cases worldwide. HIV infection leads to the disease-acquired immune deficiency syndrome, which leads to a reduction in the immune system, as the name says, and death from this actually is caused by secondary infections. So you might be surprised, but in the United States, we've only actually had 700,000 deaths since the beginning of, I guess, what we call the global pandemic of HIV. 700,000 sounds like a lot, but we compare that with COVID-19. In 2 years, we did well over 1 million deaths. HIV, we've had very good success at prolonging people's life because we've been able to find a large number of drugs to block the viral replication. So in other countries, in other regions, particularly Africa, HIV kills many more people because they don't have access to these drugs. HIV, remember, has a spike protein that binds to the CD4 receptor on helper T cells. So we talked all about those in the last chapter. There's also a secondary co-receptor called CCR5 that's important for this binding. And there's some interesting research. People that are mutants that are lacking CCR5 are actually immune to HIV infection. So we're wondering if we can block the binding process to maybe block the viral entry. So there's a target of medications there. When HIV gets in, it disrupts cell communication and the immune system, and the DNA integrates into the host genome. Once it's there forever. That's really the scary part about HIV, right? It integrates with our genome, and we do not have a way to remove that. There is a science fiction-y hope that one day we will have a technology that can go in and specifically cut out that piece of DNA and remove it. We do have some technologies. You've probably heard of CRISPR-Cas9 for genome editing. Theoretically, that could do that, but it's a long ways off. So it's going to be years before we have a way to do that. But whoever figures that out is going to be very famous and make a lot of money, because that is the real goal, to curing HIV. Otherwise, all we're doing is preventing viral replication. We are not curing it. One protein that HIV makes is called Nef. Nef inhibits apoptosis. So apoptosis is programmed cell death. Usually when cells get infected with viruses, they go through programmed cell death to destroy themselves. That destroys the virus. So HIV prevents that from happening, prolonging the cell's life, meaning more viral copies are made. Eventually, those infected T cells will be destroyed by cytotoxic CD8 T cells. Over time, that's going to lead to a diminishing of the immune system. So, right, we have an initial infection. So on this, we have T cell count. In orange, we have HIV RNA copies. Initial infection, viral copies go up quite high, and there's a dip in T cells. Then it goes into kind of what we call clinical latency, where viral level drops quite low, and T cells recover some, but they continue to decline over a period of years. Eventually, they will decline to a point where the immune system is no longer functional. At this point, RNA levels of the virus, so basically viral copies, spike, and other infections start occurring. Pneumonia, things like that. We talked about toxoplasma. That's a common one as well. It's those other infections that might lead to death. So we generally try to keep viral numbers low by using combination therapies. We target something like the reverse transcriptase gene, which is the retro part of the retrovirus, right? We have several other proteins that we're hopeful of targeting. We talked about Nef, that reduces apoptosis, right? There's also ones called Tat, transcriptional transactivator, which kind of accelerates HIV transcription. And we hope that we can maybe produce a vaccine against this molecule. It decreases MHC I production. So that disables the immune system, right? Remember, MHC I is the signal on cells that says, hey, I'm infected. I need you to come destroy me. So if we could, TAT blocks this. If we could get rid of TAT, we could maybe kill those cells much more effectively. Okay, here is another example. We'll talk about a papillomavirus here. So we have Janine, she's a 45-year-old migrant crop worker from Belize. Over the past 2 months, she has noticed abnormal vaginal bleeding and a foul-smelling yellow discharge. So that's not good. She goes to a clinic and has a pelvic exam. And she's hoping that it's just, I don't know, something she can take some antibiotics for and it will cure her. Unfortunately, the doctor performs pelvic exam and does a pap smear and notices the advanced stages of cervical cancer. So a pap smear is gathering cells, particularly cervical cells, and then they're looked at under a microscope. And if you're trained in this, you can notice that these cells look pretty normal, but this one is very abnormal. It's got all the weird things going on. It's got multiple nuclei and stuff. Pap smear is looking at the cells, looking to see if there are cancerous ones in there because they look different than normal healthy cells. They also did a DNA test for human papillomavirus. Janine has had multiple sexual partners over the years, and she also recalled a wart-like growth on her genitals about 20 years ago. So it must have been memorable, but it went away. She was embarrassed by it, but it went away, so she didn't think anything of it, even though she clearly remembered it, right? So the pap smear comes back. We have late-stage cervical cancer, and DNA test confirms human papillomavirus. So here is the cancerous cervix coming up, just a little bit of warning. What happens here? She hadn't been going for routine pap smears and was sexually active. That increases her risk. I'm not telling you that that's demonizing it, but it does increase the risk of transmitting HPV. We've talked about HPV. We have a vaccine against it. So it's very important for young people to get that before they become sexually active, because once you have HPV, you can't get vaccinated against it. HPV causes abnormal growth of cells. So here you can see cancer, and remember, cancer is abnormal cell growth. So the cancerous growth in the cervix here, whereas in a healthy cervix, it does not have cancerous growth there. There are many different types of HPV. This is just one of them that causes cervical cancer. Other types could include warts on the skin. So what HPV does is it infects epithelial cells, and it actually increases their rate of division. That's because each cell that is infected, if it divides, now you have 2 cells that are infected. And if those divide, 4 cells, so on and so forth. The way HPV does this is it makes one protein that we call E7 that binds to a normal human checkpoint protein. So normally, the human cells have many checkpoints that prevent cell division, right? We don't want cells to divide abnormally because that can lead to cancer. So there's a lot of checkpoints to say, nope, it's not time to divide right now. HPV goes in and destroys that checkpoint, opening the floodgates to cell division at this point. And so that leads to warts or potentially cancerous growths occurring. Okay, one last type of virus that we're going to talk about. Luckily not as deadly or cancer causing, but it can cause lesions on the epithelium. This is herpes virus. So there's actually 2 strains of herpes virus, HSV1, which causes cold sores, commonly found in the mouth, and HSV2, which is genital herpes. So after primary infection, the herpes virus goes into the cell and becomes what we call latent. It tends to infect nerve cells and white blood cells. So basically the viral DNA circularizes and exists in a little vacuole inside the cell. So it's not integrated into the genome, but it is in the cell there. and it rarely integrates with the host genome. So it's inside the cell, hidden from the immune system. So herpes viruses can emerge from these latencies when they get reactivated. We know the stimuli that tend to cause this reactivation, but we don't always understand why, what the signals are really doing. So we know that UV exposure can do this. Emotional stress, which is interesting because how is that reactivating a virus? That's something interesting, I think. Fever, immunosuppression. So you might know they're called cold sores because when you have another infection, oftentimes they reactivate. And what the virus does is it makes tiny RNA molecules called small RNAs or microRNAs. These don't code for proteins. They actually match up to other RNAs like our mRNAs, and they bind to them and destroy them. So in this way, the microRNAs block the host cell's apoptosis program. So they keep those cells around longer, keep them dividing. So herpes virus is very interesting. This latency is problematic. It's hard to get rid of them because they stick around for so long. All right, finally, this one I think is super clever. There are some viruses that evade the immune system by preventing the production of MHC1. Remember, MHC1 signals that there is an intracellular infection. Our body's not that stupid, though. It realizes that if a cell doesn't have MHC1, something is off, and that might be a sign of a viral infection. So viruses evolved this thing, we've evolved a defense against it. So here's the thing. Natural killer cells will go and kill those cells. So some viruses have developed mimic MHC1 proteins. So they make a protein that looks kind of like MHC1, but it binds to certain cells and prevents them from destroying the cell. So they evolved something, we evolved something, they evolved something. It's this constant battle. And this is the thing about pathogens, right? We're constantly evolving immune defenses, and then they evolve against those immune defenses. Fascinating subject. All right, let's finish up with a little review question here. HIV negative factor, Nef, increases HIV virulence by A, increasing T cell apoptosis, B, preventing T cell apoptosis, C, blocking natural killer cells, or D, reducing HIV replication. Pause the video, think about this one. Okay, hopefully you spotted the one that makes no sense. HIV would not be more virulent if it reduced its own replication, right? So that one's out. We didn't talk about natural killer cells with HIV, so hopefully you eliminated that one. That leaves two apoptosis ones. So remember what was happening in apoptosis. This is programmed cell death. Preventing cell death leads to more viral particles being made because the virus needs the cell to reproduce. So preventing T cell apoptosis is the role of Nef here. Okay, we talked a lot about influenza versus rhinovirus, why we tend to get reinfected with different viruses, and why some are more deadly than others. Antigenic shift, that was really important in influenza, rearranging the segments of the genome. Drift is a slower change, but it can lead to new viral antigens. And co-infection of influenza can lead to that shift. And we did talk about some that could become latent. Herpes viruses particularly have latency, which makes them hard to get rid of for the immune system. Okay, that's it for chapter 18.